Welcome everybody to the second session on toxicology here at the pesticide conference. Welcome to everybody, welcome to the speakers. So I would like to ask the first speaker to come here. Sofia Morcel, she's working at CEO here in Spain, and she's going to tell us about how to work with farmers to protect bird life. Floor is yours.
Thank you very much. Thanks to all the organization for this forum as well. As Daniela said, I'm Sofia Morcel. I work for CEO BirdLife. It's an NGO that works with nature conservation in Spain. On this session, I want to talk about pesticides and wildlife-friendly farming. As we all know, pesticides, agrochemicals, used to protect humans from various diseases and are also known by their ability to cause a large number of negative health and environmental effects. We know that some pesticides disrupt food webs, killing plants and insects and removing weed seeds from the environment, which may be essential food or habitats for some bird species, and so represents a high risk for their conservation. On CEO BirdLife, we are very concerned by the environmental impacts of the widespread use of pesticides, and we believe that if they must be used, it should be done subjected to appropriate scrutiny in a targeted way and with respect to the environment. On the other hand, we are living in a biodiversity crisis, and there are multiple threats facing our wildlife, including habitat loss, pollution, and climate change. We are with around 1 million species facing extinction worldwide. Population of European farm birds having declined by 4% since the 90s, and dramatic insect deadliness right across the globe. So we think that governments need to step up their support for farmers to facilitate this at a larger scale and protect people and nature. But how much pesticide is used in Spain in agriculture? The vast majority of pesticide in Spain is used in agriculture. This table, it's not working, indicates the available data for overall treated area of arable crops in Spain per type. The table shows that the number of hectares treated with pesticides have increased by 4% in the last 8 years. That's not a lot, but if we go to this other table, shows the overall amount of pesticides per hectare used in arable crops at a national level between 2011 and 2019. This data shows how pesticides per hectare in Spain increased by 57% in the last 8 years, which is quite a lot. And also, if you check the green vegetables, it has increased. You see there that it has increased a lot in the last 8 years. So one of the case study that we want to talk here is about two different bird species, red-ledged partridge and gray partridge. On the first experiment on red-ledged partridge, previous studies has shown that chick mortality was a key factor, and that this was related to the quantity of insects they have available to eat. The number of chicks in each brood have, when the field were sprayed conventionally, conventionally with pesticide, compared to the fields where a 6-meter strip around the edge of the field was left unsprayed. On another study, another experiment with grey partridge, the test done with some scientists wanted to know whether the herbicides and fungicides used on cereal fields reduce the survival of grey partridge chicks. They saw that the elimination of pesticides requires activations of various detoxification systems in an organism, and that energy and resources allocated to biodegradation of pesticides cannot be allocated to other functions such as growth. Compared to, so they use two different groups of chicks. They use one group of chicks fed with organic grains, and another one fed with conventional grains where they were treated with pesticides. On the one that they fed with conventional grains, they saw that compared with organic pairs, conventional pairs yield smaller chicks at hatching that had a lower body mass index at 24 days old. But additionally, these chicks also displayed lower hematocrit. So on conclusion, they saw, the scientists saw that the chicks that were fed with conventional grains where pesticides had been used, their body mass were lower, their shape and their health was also lower than the ones that were fed with organic grains. On this other case study, I'm sorry because I don't know why, but the letters are not very well shown here. It must be about the type of letters that I'm using. On this European project we are currently working on, that is known as Olivares Vivos, is a LIFE project. We are working with farmers using environmental friendly systems. We've seen after 5 years of projects that farmers they had some anxious moments when they first started because they didn't know very well how the project will help them to have better crops and also a good product in the end. But they saw that the crop yields are a little lower now, but the yield losses are more than made up for by the reduction in how much they spend to produce the food. And what is more important is that their farm now is more resilient to weather and also to market fluctuations. And also one of the important factors that we've seen after this project, known as Olivares Vivos, is that we have more pollinator species, we have a better pest control of the crops. Also, the farmer have to invest less on pesticides, and also one of the farmers that we are working on, they recently won an award, an international award for olive oil. So the company increased its international prestige as well. So we have different conclusions here. The first of it is that BirdLife is very concerned about the environmental impacts in the widespread of pesticides. We believe that if they must be used, it should be with care, with a targeted way, and with respect to the environment. We are calling for a national pesticide reduction target to be implemented in Spain. This should include the creation of an independent organization that encourages farmers improving the natural health of our countryside. And this organization should be implemented with the help of environmental NGOs, such as BirdLife, and should promote initiatives such as free training systems and gathering events. We need also a better support for an integrated pest management approach, where pesticides are used only as a last resource. And in this regard, pesticide registration is an important step in the management of pesticides. We've seen in Spain that some of the pesticides that have been sold are not yet registered. And this, if they are registered, enables authorities, primarily to determine which products are permitted to be used and for what purpose. And it also makes possible the control over quality. Also, we must recognize the important role that the healthy, thriving ecosystem can play in supporting sustainable farming. And pest predators, pollinators, wildlife in the soil can all help to grow food and sustain long-term yields, while continued reliance on pesticides threaten long-term sustainable food production and a healthy environment. And finally, we encourage environmental stewardship agreements, not only in Spain, but in the whole Europe, with the main aim of reducing the risk and the use of pesticides on the environment. This could be an effective way to support sustainable farming. Thank you very much. And also, if you have any questions, you can ask me now, or either you can email me on that email that I'm showing you on the screen. Thank you.
You said it would be important to promote IPM. Now, there's always two ways how something can move. You either push it or you pull it. In Switzerland, we have a lot of success with organic and also IPM, but the big reason is in my country, we have two very big food companies, food retailers, Migros and Coop. And Coop 30 years ago made it a strategic decision that they want to promote organic production, and they have about 40% of the market share, so that was an important decision. And Migros was then forced 10 years later on, forced to take a similar decision, saying that we will not go organic, but we will go IPM. So I don't know whether in Spain you have similar retailing giants, but maybe having them into the boat and saying we give to farmers a better price, we support them in transferring to organic production, you would have an important ally who could help you promoting what you try to do.
Thank you. Yeah, we have big retailers here in Spain, but I think, I don't think, as far as I know, I don't think they have implemented those changes. And maybe my organization needs to work together with those retailers to make those changes in the future.
Just do it. Maybe it doesn't work first time, but fifth time it will work.
Yeah, I think that's a good idea. Thank you very much.
Thank you. I'm impressed by the presentation. You are very rational and very correct in your analysis. My question is, in Spain, this all makes sense, but what are the barriers that are preventing its dissemination and uptake by farmers? What do you think are the biggest barriers to that?
I think one of the most important barriers that they have is that they don't have the information available. So that's why we want to open farmers to a kind of new NGO or I don't know if the correct possibility would be a new NGO organization, but something where they can all be connected, they can get information, and they can change to use less pesticides in the future. Also, I think that some of the farmers right now are also afraid of taking that change in the future. But I think also one more time is because they don't have enough information to take that step forward.
Now I would like to introduce our second speaker, Anna Maria Iannetta from University of Teramo in Italy.
Good morning. The study that I present today concerning the toxicological evaluation of glyphosate in zebrafish early life stage. And glyphosate is a systemic and non-selective post-emergency foliar herbicide, and is today known as the most widely used herbicide worldwide. This herbicide operates at the level of APSPS enzyme, inhibiting him, and the inhibition of this enzyme determines the phytotoxicity of the compound and destroy the plant by three ways. The first way is the loss of essential amino acid. The second way is the alteration of chlorophyll photosynthesis. And the third way is the accumulation of toxic products. The aim of this study is to investigate the potential effect of the development of zebrafish early life stage, and we use this in vivo model because this is an excellent tool for experimental studies that concerning the toxicity of environmental contaminants. Zebrafish early life stage used in the experiment were obtained from the University of Teramo facility, and adult wild-type AB strain zebrafish were bred in a recirculating water system with a light dark cycle. The day after, the afternoon before the spawning, the groups of male and male were placed in a breeding tanks, and the eggs were collected the following morning. The fish embryo acute toxicity tests were performed in according to OECD guidelines, and the aim of this test is to evaluate the acute toxicity of glyphosate in zebrafish early life stage. The tested concentration were 25, 50, 100, 110, 125 milligram per liter. And negative control and positive control were also tested. 3 replicates were performed, and the selected embryons were placed individually in 2 ml of test solution in each well. And 20 embryons were tested for each treatment. Zebrafish early life stage were daily observed up to 96 hours post-fertilization with the inverted optical microscope, considering for lethal alteration, the coagulation of embryo, the lack of detachment of the tail from the yolk sac, lack of the somit formation, absence of heartbeat. And at the end of the exposure period were calculated the lethal dose 10, lethal dose 20, and lethal dose 50. The sublethal effect and the histological analysis was performed. For the histological analysis, survived larvae at 96 hours post-fertilization, treated with all concentration, were fixed in 10% neutral buffered formalin for 2 hours, and after the thickness section were stained with hematoxylin and eosin, and then visualized by light microscope. The results of fish embryo acute toxicity test were obtained by ToxRat software, and at the 1960 hour post-fertilization, the LD10 was 85, the LD20 was 96, and the LD50 was 122. And regarding the toxicological endpoints, there are different studies in agreement or not with our results. In fact, in this first study, the lethal dose 50 is lower than our results. This because in this study they use different chemicals. But in the second study, at 96 hour post-fertilization at 50 milligram per liter, the LD50 is about 20% in according with our results. In addition to toxicological endpoint, we evaluated also the sublethal alteration. In fact, but only for certain concentration. And in fact, from the concentration of 100 and 110 milligram per liter, the larvae showed a pericardial edema and impaired blood flow and blostasis. In addition, a great number of zebrafish larvae showed a smaller head than the negative control. It's possible to observe in the figure C the normal size of the head and the pericardial region, and in the figure A and B the small head and the increase of the pericardial region in larvae that was treated with glyphosate. Regarding the cardiotoxicity, in according with several authors, this alteration can be related to the dysregulation of myocyte enhancer factor 2 that is involved in the development of heart muscle. And regarding the smaller head that showed the larvae exposed of glyphosate, this alteration suggesting a neurological involvement. In fact, according to several studies, this alteration can be related to reduction of essential genes for the normal development of the central nervous system, such as PAX2, PAX6, OITX2, EPHA4. In fact, the aim of the future will be to evaluate the alteration of the regulation of these genes. For the results of histological analysis, this confirmed the results of FIT test. In fact, 18% of embryo treated with glyphosate at 100 and 110 milligram per liter showed high pericardial edema and diffuse hepatic microvaculus that is possible to observe in these two sections. In conclusion, in this study, we confirmed the toxicity of glyphosate in zebrafish early life stage at the tested concentration, and glyphosate has been proclaimed safe for the human and for animals because they don't have the target APSPS enzyme. But several studies demonstrate that many microbes have the shikimate pathway, and this aspect can be represent a risk for human microbiota. Finally, the aim of future study will be to evaluate the long-term effect of glyphosate. Thank you for your attention.
First of all, congratulations for your work. We have one doubt. How did you introduce to the fish this glyphosate toxicity? How do you conduct this first test to include and to introduce the glyphosate in the fish? Okay, so first of all, what you can do is to introduce it in the egg to find other studies.
In the first, we search in the literature other study when evaluated the acute toxicity in zebrafish. That is not at present. You do a ranging test when you taste a different concentration, and after you do the fit test. It's the method is this.
Professor Emeritus Ahmad Madavi, he's going to talk to us about highly hazardous pesticide use in Iran compared to the EU.
After many years of work on pesticides, in recent years, there are lots of discussion about the new or a particular group of compounds called highly hazardous pesticides, that they have big and both acute and also chronic effects on different life system. And so we started wide work since some years ago to manage to maybe eradicate, or not eradication is very difficult. I think this one, okay, let's go. This is the topics that I'm going to cover in this short time now. There are, many problems with pesticides that, especially with hazardous pesticides, and we were discussing about these compounds under the SciCam discussion since we were in Cambridge in 2019, before Corona. Then later we had to sit by the computer and go every day to talk about these compounds and how to manage them, really. It was a difficult one. There are many problems, as you see. We dealt, because I am basically insect toxicologist, we went through the life of insects in Guelph, Canada, and other places during my PhD, even to the depth of the Darwin, and to see really what are these creatures since Precambrian era, about 300 million years ago, and their relations with plants. They are not going. We have less and less population of insects that is supposed to be 5 times of human mass population on the earth. But there are many families are whole disappearing from the planet. And that's a big, alert and danger that we have to consider it. There are, honeybees that is been discussing a long time about it, and pollinators. The bad problem is that with pesticide, dangerous compound like pesticide, in developing countries, the management is different. In developed world, they are using lots of pesticide. The management is good, but we have so many people get killed because of bad infrastructure. We call it bad infrastructure. And so it's causing lots of damage to people, environment, and wildlife. That's something that we have been going to many conferences and discussing about this since about 20 years ago. And there are big disasters, in Bhopal. Also, we had disasters in Iran. It happened in Peru. It happened again in Bihar, India. Children killed by this very dangerous compound, highly hazardous pesticide. And there are global agencies, FAO and WHO, they are trying to manage, but they really have to consider, many times I mentioned myself in our discussions, they have to consider the problems of these compounds in developing countries separately. Separate even regulation and management, because it's different. So because of it didn't work. And every day, as I mentioned yesterday, there in another session, they are increasing these compounds every year, more and more use. There are lots of discussion about highly hazardous pesticides now, because they are known dangerous materials in 5 groups. And this is where we discuss in SciCam, under this SciCam. For me, it's a long time, really, about 50 years work on pesticides, since 1972 that I'm working. Now, see, all are working, and we are aware about all these works, about the pesticide and gender issues, ladies, women. Even though they say that in men, sperm count is getting low, but really the danger for pregnant women, we had a different discussions, the ladies group, that one good one in Germany, Dr. Hemmati and others, there are now very good discussion that I was also part of that, and we were following that. I did also high exposure to these compounds myself. I got 2, 3, 4 times getting really to die. When I was starting to use and work for my research, it was about, as I said, 50 years ago. That was 50 years ago, 10 years after the Rachel Carson, gave alert to the world about these compounds. But still, because I was in Iran, in many developing, there was nothing about environment that time, nothing. And we were exposed, because I was doing lots of experiments in garden and crops, and working for professors that had relation with companies, not now. Companies had a very, very good activities in Iran. Good, it means wide, but with dangerous material. So there was, in Iran, we also here in cotton fields, that I was there. Big planes, hundreds. They were Bulgarian, they call it that. Really this side envergure of the wing, and hundreds of them every day in the northern Iran by the Caspian. They were polluting the whole thing, all types of pups. Aldrin, dirdelin, that we called it HEO, a compound turbidon. It was a mixture of the DDT, paration and DDT, paration, DDT, and toxafen. All type, and now the soil is there polluted, very polluted. That's what really I wanted to tell these companies maybe to have a work, is the northeast of Iran near Turkmenistan. I was there that time. I'm talking about maybe, and later of that, maybe 45 to 50 years ago. There are lots of pollution. I had some talks in recent years in China and other places about wetlands. I am a wetland person also with Ramsar. Because I lived and worked about 20 years in Mazandaran by the Caspian. Mazandaran is the place of Ramsar. The wetland is under the name of Ramsar. The very beautiful city that Ramsar Convention started about 1972, more than 50 years ago. So we were just working and trying to see how much damage from the agricultural rice paddies come to the wetlands, a lot. a lot. And birds, we were related to the migratory birds also, a convention of CMS that I talked for it in Toledo here, very other beautiful city, about the lady talks about the wildlife exposure. I talked about it, that how much pesticides really we showed them are dangerous for birds. We have a city in the Mazandaran, it's called Feridunkelar. It's called all over the world as the slaughterhouse of migratory birds, because migratory birds come in the winter and they all get exposed and killed. 10 million comes to Iran and 5 million goes back. And it's 10 years, 20 years that we are giving to the environment. But no, you know in Iran what's happening. I'm not scared to say that, They release everything, doing nothing, just religious thing and everything. So no, really in a bad situation. I had many, conferences. There's few of them that we thought, and this is also a Toledo talk that we did there about the exposure of this. The insects are going. Insects themselves as for example, food of the birds. And even that one is affecting on the population of birds. Many of these pesticides, I worked with them during these 50 work, different work, lab work, in the analysis. I stood 10 years by GC analysis. Different GCs I worked. We did different works on the experimental work in the field. But these are all, this diazinon is supposed to go, but it still come back. You can find it always in the field. And very, very dangerous material, aldicarb, it's chemic, 2 milligram per kilogram. We worked very good in Guelph that I did PhD and later other places about the real good that I asked the lady about acute toxicity testing, how to analyze it. We wrote a program that later they used it in US even. So insects themselves are going, and there's a big discussion about it that I'm following, that pollinators are going. If preventing poisoning, it came back, I think. See, this is what I asked today, axial thickness, that it is my Facebook, just is a picture of my Facebook, that is all about this, my Facebook, since many years ago. Yeah, these are going and coming back in Iranian market, as well as in the whole MENA. We try to concentrate on the Middle East and North African countries. That it is now the place of those dust, whole. Go to the Google and you see the color is different. We have all dust cover, and many people believe that it's going by 30 years, there will be nothing. No Saudi Arabia, no Iran, nothing, because of the desert certification that now is going very bad. And nobody, in other countries, Saudi Arabia is doing very good now. They're doing good plan to just greenery there, but nothing in Iran, unfortunately. I introduced the neem tree. This is Persian louse powder, but this is neem that I introduced it from the Persian Gulf. I didn't discover it, it was there. Somebody 200 years ago brought it to Iran, Persian Gulf Islands. And I cultivated, I planted thousands of neem tree. That is a very good source of natural pesticide. This is Persian louse powder, that it was the first pesticide on the planet 2,000 years ago. Go to the internet, please, you can find it. Persian louse powder. Iranians were exporting louse and cooties, and the problem today again. It is coming back. Anyway, we found that regulation stops at the border. And really good regulations that exist in Europe and North America.
Could you think about the time?
How much?
Can you come to an end slowly?
Is closing. Okay, thank you very much. It's not coming to developing countries. And we have bad situations really. Because of what I recommended always is certification and licensing. The best recommendation that in our discussion that I mentioned with South Africa, Cape Town University is going every week. I always recommend we need certification and licensing safe system in developing countries. We discussed a lot with FAO about these things, but hopefully that these things happen soon. In the Toledo, we discussed about the second generation anticoagulant rodenticide that doing a lot of harm to, other rodents and other birds also. That's a dangerous situation. This is a long work for me, and these are some of the recommendations that I made, and you can find it even online if you go searching my name or other things on Google. Some of these recommendations from our discussion, SciCam discussion, are there about highly hazardous pesticides in the Pesticide Forum. Also chemicals in product, as well as lead poisoning, is more about lead paint. But also lead poisoning, we found that in our wetlands, lead are doing a lot poisoning to birds because of bullets. They hunt and that things. I'm sorry that this kind of discussion needs more time really, but these are something that my question to the SciCam discussion, that what we need in developing countries really to make the situation a little better. Mr. John Wittgen is a great man. One problem is that FAO, WHO, they are not giving us opportunity. We know the problem in developing countries, but they are not giving us. Right now, tomorrow I was supposed to go to Nairobi, in that science policy. But they always go to the government people, those ones in Iran that praise only, or other places in Middle East, or other places. They have to give opportunity to the people that are involved with this science. Anyway, this is John helped me, a great man. We were communicating since 20 years ago. And 8 years ago, I couldn't come to this city. That I found later why this city is a beautiful place, because I'm a kind of photographer also. And thank you that he made it possible for me. There are lots of other things that here, at least a picture maybe from Iran. No, that's this is my civil society, and this is where I did retire myself 20 years ago, because I was not able to do both religion and science. 20 years ago, I did retire myself from here, and I'm doing just global things now. Thank you very much. Thanks. Thank you.
I would like to welcome another speaker from Italy, Marco Minacori from University Sapienza in Rome.
Good morning to everyone. I'm Marco, and this morning I would share with you some cellular results that can, I hope, that can provide the molecular basis to hypothesize a human body remediation for exposed population to pollutants, and especially exposed to beta HCH. Okay, in Italy, we have 42 contaminated areas called contaminated areas of national interest. And one of these is the Sacco Valley on south of Rome. And the environmental problem of this valley emerged in 2005 when 25 cows died after drinking water from the Sacco River. And after the veterinary investigation, the monitoring system, national monitoring system on chemical residuals in food found a lot of pesticides, both in the blood and in the milk of cows grazing in the Sacco Valley. Among all these pesticides, there was the beta HCH. Probably a lot of you know about beta HCH, but shortly, beta HCH with the alpha isomer, epsilon, delta isomers is a bioproduct of the synthesis of gamma isomer, better known as lindane. All alpha, beta, epsilon, delta are waste. And in Italy, we stopped to product in the Sacco Valley. We stopped to product lindane in 60s, but its legacy is today present with both in the soil and in the populations with the fossil isomer, the beta HCH. Okay, this is another picture of Sacco River that can help you to better understand the real condition of this river. This picture was taken in 2018. In the same year, my research group published the first paper in which STAT3 protein, an oncogenic protein, was individuated as an hub protein of a signaling pathway triggered by beta-hexachlorocyclohexane. In 2020, another paper from my research group in which were individuated a number of beta-hexachlorocyclohexane cellular targets. And in 2021, we demonstrated that beta-hexachlorocyclohexane can drive all three stages of carcinogenesis: initiation, promotion, and progression. Yeah, from literature, we understand that the scientific world interest is focused especially on the environmental remediation from beta-HCH. But what to do with contaminated people, contaminated population who lives in contaminated areas by beta-hexachlorocyclohexane and that have beta-HCH in their organism? Well, we think that to manage this level, we need of something that can modulate the beta-HCH activities, cellular biological activities, something of affordable, quickly available for all, and with a low impact on the environment. For this reason, of course, the answer to all these characteristics, we found the answer of this to all these characteristics in natural compound, and especially natural compound present in diet. Okay, so according to our data, our previous data, we know that beta-HCH exerts a lot of cellular activity. We know that it has an impact on cell cycle and apoptosis. It induces the DNA damage. It induces the oxidative stress with the switch of the energy metabolism. We know that it is an endocrine disruptor. We observed the activation of androgenic receptor. And it is an activator of oncogenic pathways. For example, the activation of the oncogenic protein STAT3. So we searched in literature natural bioactive compound able to modulate all these characteristics, all this cellular activity. And we found a lot of natural compound, for example, lycopene, tocopherol, tyrosol, hydroxytyrosol. All these compounds and others, all these compounds are present in two important elements of the Mediterranean diet and also common worldwide, that are tomato and olive. So, for our experiment, we decided to treat cells with a supplementary food, a patented supplementary food, composed by the 98% of whole atomized tomato and the 2% of olive meal water. And, we prepare a solution from this patented supplementary food that in our experiment we called TOBC solution, tomato and olive bioactive compound. This solution is enriched of all these compounds from tomato and olive. The experimental concentration in our experiment, the experimental concentration of beta HCH was extrapolated from an epidemiological study on Sacco Valley, on people who live in Sacco Valley. This is the experimental concentration of a TOBC solution, and the cell line that we used in our experiment is LNCAP, that is a prostate carcinoma. Okay, this is the first result, and it is an MTT assay. Is an assay to evaluate the cellular proliferation. Control is represented by untreated cells, and if we treat for 48 hours cells with beta HCH, we can observe an increase of the cellular proliferation. But if we pre-treat for 3 hours cells with TOBC and then for 48 hours with TOBC and then beta HCH, there is a drastically reduction of cellular proliferation. Then we observed the activation of oncogenic pathways. STAT3 protein is an oncogenic protein, cytosolic oncogenic protein. After the activation, with the phosphorylation on tyrosine 705, there is a nuclear translocation of this protein, and in the nucleus it is transcription factors, a transcription factor for a lot of genes involved in the cellular proliferation, migration, invasion, and this is a Western blot with the relative densitometry, and on the first line there is the activated STAT3. In the control there isn't the activation of this protein. With the treatment of beta HCH there is a STAT3 activation, and if we pre-treat cells with TOBC there is a drastically reduction of this protein, of the activation of this protein. With this immunofluorescence, we observed the same thing, but we observed the nuclear translocation of this protein, because in the first column there is the fluorescence of nucleus, in the second, the fluorescence of the protein, and in the third, the merge of the two columns. In the control, there isn't nuclear translocation. STAT3 is around the nucleus. With beta HCH, there is the nuclear translocation, and this nuclear translocation is reduced with TOBC. Here, observe the other two receptors, androgen receptor and arylic hydrocarbon receptor. Also in this case, the activation of this receptor induces the nuclear translocation, and with beta HCH there is a nuclear translocation of both receptors respect to control that are untreated cells, and in the co-treatment this nuclear translocation is reduced. Then we observed the reactive oxygen species induced by beta HCH, and with TOBC there is a reduction of these ROS species. Then we did another assay that is a comet assay to evaluate the DNA damage. After the DNA damage, there is a fragmentation of DNA. And with this assay, that is an electrophoresis to DNA, if there is a DNA fragmentation, we can observe a tail. If there isn't DNA damage, we also observe only the head of the comet. In fact, in the control, there is only the head of only the head of this comet. In beta HCH, we can observe the tail. It means that there is DNA damage. In the co-treatment with TOBC and beta HCH, there is a reduction of this tail. Then we observed the cellular apoptosis, and beta HCH doesn't induce apoptosis in cells, but in the with the co-treatment with the TOBC, we have a cellular apoptosis of these cancer cells. The last experiment is a colony formation assay, and in this experiment we observe the clonogenic activity of cells, and the more is the number of colonies, more is aggressive the phenotypes. And in fact, if we treat cells with beta HCH, there is an increase of colony number. This number of colony is reduced by the pretreatment with the TOBC. To conclude, in our opinion, the environmental remediation concomitant with a continuous health surveillance can be enforced by population remediation strategies. In this context, proper use of standardized, improved functional food may be an attractive strategy to implement the protection of exposed population. From our data, TOBC complex of micronutrients can reduce the genotoxic and carcinogenic activity of beta HCH. This in vitro study provide, we hope, the basis for exploring new strategy for the protection of exposed population. Thank you for your kind attention.
Thank you.
Hi, thanks for the interesting presentation. My question is, have you got evidence of elevated levels of cancer or DNA damage amongst the population that is exposed to the beta HCH? And the second part of the question is, in Italy they eat a lot of olive oil and tomatoes, so maybe they're already protected.
We haven't evidence in population, unfortunately, but the Italy situation is just a little bit strange. There is a good management of this situation, unfortunately, and so we haven't a lot of data on populations. We think that tomato and olive can be a good solution, but are also common, especially tomato is very common all over the world, so I think that could be a different way to observe the remediation. I don't know, because it's really important, the remediation of the population, the populations, because compound like this has a long half-life in body. So what should, environmental remediation is important, is very, very important. But what to do with people who already have and have to stay with these friends for a lot of years?
So am I audible? Hi everyone, we are from Osh, Kyrgyzstan, and our first presentation is about the use of therapeutic agents derived from the plants and fruits growing in Kyrgyzstan for the elimination of OCPs from GIT tract of nursing women. POPs, including the most common and dangerous OCPs, so is a problem that remains pressing in rural areas. And OCP exposure is linked to higher rates of various pathologies of pregnant women, fetuses, newborns, and infants. So in southern Kyrgyzstan, OCPs are the major cause of a number of pathologies, which are including as cancers, hepatitis, infant encephalopathies, dysbacteriosis, immunodeficiency, and so on. It's well known that the pectin substances are used to remove the ecotoxicants. So for the prevention of the above pathologies, taking into account that like most of the OCPs, like probably 100%, are taken into the body through the GAT. So there is only one way to eliminate OCPs from the GAT, and this is a search of therapeutic agents for their elimination from the body. Because as you know, the pesticides are used to be accumulated in the body and they cannot be removed by the natural way. So the breast milk samples were collected from the women who contacted us about illness of their children, like from the maternity hospital and the women themselves. So all samples were collected with the written consent of participants according to all ethical standards. Special attention was given to the cases, and there were most of them when the examinations were carried out at the request for the examined woman. Like since we covered all of the expenses for the examination, further conducting treatment, aimed at removing of OCPs from the body of nursing women, mothers, with the use of therapeutic agents. So we obtain it from local endemic plants and their fruits which possess sorbent and detoxic properties. So, therefore selected the woman who came to us on 28th, third day after the childbirth, after the labor. So the concentrate was used in the form of tea daily, like 2 teaspoons, which means 15 grams per 300 milliliters of boiled water, brewed in a thermos and administered during a day. So a golden root tincture, analogous to ginseng, produced by the Institute of Medical Problems, derived from Radiola rosea, like the proportion is 1 to 10 in a 30% of alcohol solution, which grows in a high mountains region. Like the altitude is about from 2,500 up to 3,500 meters above the sea level. In Chongalai district, this is the district of the Osh region in the Kyrgyz Republic. So the tincture was administered by 25-30 drops twice in a day. The course of treatment was from 10 to 12 days. So this is the program which we use for the processing of the information. So in table 1, you can see the results of toxicology examination of the breast milk, which were taken morning or anterior and posterior, which mean residual portions of breast milk. Like from, in total, we have collected from 82 nursing mothers. So breast milk samples, about 10 milliliters, were collected in a sterile disposable tube with mincing syringe with a lid and then transported to the laboratory in the same container. So sampling of breast milk, blood, and urine for OCP was carried out in accordance with the methodological recommendation on the gaze chromatograph, which called CVET 500M. It like certified and manufactured in the Russia. The presence of the following pesticide was determined for the HCH, alpha, beta, gamma, sigma isomers, DDT, DDD, and DDE isomers like aldrin, D-aldrin, and geptachlor. So all women under observation were allocated into the two groups. First group consisted of 27 nursing women, and group 2, which is control group, included 24 women. Women in group 2 didn't receive any treatment. In group 1, the concentrate derived from medical herbs was used to neutralize and remove the pesticides from GAT, including crushed seeds, which contains unsaturated fatty acids up to the 4%, growing in southern region of the Kyrgyz Republic and mid and high mountain areas. So they are rich in vitamins, pectins, microelements, and biological active substances. So with slightly bile, uric, and diaphoretic properties. So the following types of OCP were found in the breast milk of the woman under the observation. So in the first group, the number of samples was 27, and HCH was found in 15 of them. So the percentage is about 55%. DDE was found in 24 samples of the breast milk. So here you may see the concentration level of the each pesticide, and here you may see the results after the treatment. And here you may see the results of the second control group, which didn't get any treatment. So in the second table, you may see the level of the OCPs in the blood sample. Here is the same structure. So first group, treatment, second group, without treatment. So in the blood, of the blood samples of the first group, nursing women, was found like in 9 cases, and DDE in 15. In the control group, 16 and 12. So after the treatment, the number of detected in the blood samples was 6, and DDE was 15. Concentration. So HCH in the blood samples were detected in 9 cases, which is about 33%. And after the treatment, the HCH was found in 6 cases, like about 22%. So DDE detection rate remained at the same level, about amounting to 55, but concentration level decreased. So detection rate of HCH in blood in control group remained unchanged, amounting to 66.7, but concentration level decreased by 1.36 times. The rate of DDE detection in blood, on the contrary, increased from 50 to 66.7, and concentration by 1.15 times. So in the third table, you can see the concentration levels of OCP in urine samples before and after the treatment. The level of HCH in urine samples increased from 11.1 before the treatment to 22.2 after the course of treatment, but concentration level decreased by 58 times. DDE from 33% cases before treatment to 3.7 after the treatment, and only DDE traces were detected. In control group, the levels of HCH and DDE in breast milk samples and HCH level in blood samples remained at 66.7, and in urine samples remained unchanged. DDE level in breast milk, on the contrary, increased from 50 to 66.7. In urine, the level of HCH remained at 50%, and DDE increased from 12.5 to 16.7. So in group 2, no changes were registered. In control group, the detection of rate of HCH and DDE remained unchanged. So despite the results obtained, there are still many questions to be answered. First, a question concerning the therapeutic agents derived. These agents are multi-component. They contain pectin substance with sorbent properties, contain lipids that can absorb OCPs, antioxidant, vitamins, trace elements, and other biological active substances, which can probably form compounds with OCPs, preventing absorption from the GAT and subsequently eliminated from the body. But this issue requires studies for stool samples for OCP before and after the treatment. On the other hand, we cannot exclude a possible decay or decomposition of OCPs in GAT. So this issue requires more detailed experimental studies. So thus the therapeutic agents obtained from medicinal plants, including endemic plants and fruits, as well as a tincture of the golden root, are effective for the neutralization and elimination of OCPs from GAT, but further targeted experimental studies are needed on a larger number of patients. So that's it. Thank you for your attention.
As you said yourself, the first results look interesting and encouraging, but there's still a lot of studies to do. Do you get funding easily, or that's a major issue to find funding to move on with your research? Okay, he's saying that everything is obviously linked to money, but funding for some further studies, they have not been successful so far.
Now we're going to talk about our experience of applying the results of research and evidence-based medicine for improving the awareness, achieving compliance with safety measures, and implementing recommendations by the population living in the areas polluted by OCPs. So the problem of OCP in the South Kyrgyzstan are caused by two pesticide burial grounds left since the Soviet era. So they are located in the Suzak district of the Jalalabad region. In total, in Kyrgyzstan, we have 7 regions and 45 sites of agricultural aviation with the residues of pesticides stored there is about 183 storehouses where pesticides were previously stored. So some territories, the so-called plague zones, where the OCPs were used to fight against fleas of marmots, carriers of the plague, fields where industrial crops of cotton and tobacco used to be grown, and where the DDT was used for pest control. So here you may see the map of the Kyrgyzstan, and the highlighted area, this is the Jalalabad region. And in the southern part, which is colored yellow, I think I need to line it. So it's about like the southern part of Kyrgyzstan. It's not exact, but it's about like this. So just for you to understand. So there are about 1 million hectares of croplands in the south of Kyrgyzstan. So 60% of them were used during the Soviet period to sow industrial crops, which are the tobacco and cotton. And for the growth, for the better results, were used very large amounts of pesticides. Where at the moment, because this is the croplands, now at the moment, the population is using these areas for the growth of melons, vegetables, fruits, and other species. So here you may see the condition of the storage at the moment. Despite the fact that it was used 35 years ago, OCPs are still found in the environments. This is the same condition of the storages. So the problem of preserving the health of the population living near these places comes first. So on the backside, you may see the croplands that they are used for some kind of vegetables, like corn, for example. There you may see the plants. And one of the reasons for the ingestion of OCPs from the environment is the lack of knowledge and awareness of the population about the places where the OCPs are found and their impact on human health. Here you may see that here you may see the sorry, this is the pesticides. So as you can understand, they should not be stored like this. So this photo is like widespread in the internet, and it was in some of the abstracts, and you can see that near the pesticide storage, the children were playing, and like no one controls them because the lack of awareness of the population. You may see how the cows is feeding, and as the previous lecture from Italy were said that they like poisoning of cows which drink the water from the river. So here you may see the same, like right after the rain, when the OCPs like are moving out on the surface of the ground, and after this the cows feeding there. So in 2010, in the meat of that drums, the content of COPs was like about 0.013 milligram per kilogram. HCH from 0.005 milligram per kilogram. So out of 32 poisoned people who consumed fried liver and meat of the same ram. So the serum of 3 children showed DDT, DD, and HCG alpha and betas. So in all 3 did not exceed like less than 0.005 milligrams per liter. So as you can see, the local population cultivates cotton, maize, and the other fruits near the storage of pesticides. This is the condition of the air strip. Here in the cans, you may see like it. They were like digged out by some local people. They were digged out by some local people, and they were digged out from the pesticide storage because they don't know what they. So here you may see that near the airstrip where the plants were taking the pesticides, near the airstrip, that is the new buildings and new houses because of lack of information. How much time we have? Shall I speed up?
Yes, you have about another 7 minutes.
Oh, thank you. So to raise the awareness and executive discipline of the population living in the environmentally unfavorable places, Institute of Medical Problems, which is the South Branch National Academy of Sciences, the Kyrgyz Republic, formed a group of toxicologists, chemists, microbiologists, radiologists, ecologists, epidemiologists, and other clinical laboratory and diagnostic specialists to develop acceptable, simple, easy way to implement and scientifically based methods and recommendation with the elements of evidence-based medicine for the local population. So the group studied the effects of OCPs found in the breast milk, blood, urine, feces, semen, hair, nails, tumor, and pathological tissues, organs, and in the environment, and their effects in the examined women and their newborn and children, which was mentioned in the previous presentation. So simultaneous examination were carried out by the obstetrician, gynecologist, neonatologist, neuropathologists, cardiologists, with all of the specialties. So the data obtained were entered in a specially designed research map and Excel sheet. Laboratory specialists, toxicologists, chemists, and so on collected human and environmental samples for laboratory research. Environmental experts studied the presence of active factors Epidemiologists, the disease incidence of the population depending on pesticide loads and other environmental factors. Clinical specialists carried out preventive examination of the population. Group 3 includes 17 women living in the Aragón. It's still Osh region, but it's on the southern direction, formerly cotton growing region. So the population grow cotton and are engaged in cultivation of vegetables, melons, legumes, fodder crops. Because of like better climate, the farmers in this area, they may get the fruits and vegetables like till twice in a year. So group 4 included 24 women living in the cotton growing zone in the village of Burgundy, Nokian district. All of this is the Jalabat region. So in the subgroup A included 9 women living in the village of Urujar, located like 1-2 kilometers from the unpaved agricultural airport, which functioned until 1985. Then it was turned into an arable field where cotton was growing until 1994. The population switched to cultivation of fruits, vegetables, gourds, fodder crops, and so on. So in this area, the surface of the earth has the slopes. So the soil and OCPs are washed away by irrigation water and rainfalls. Subgroup B include 24 women living in the village located at a distance like of 4 to 6 kilometers above the Agra airport. So in this study, the following types of OCPs were found in the breast milk, HCH, which are like alpha, gamma, beta, DD. So the highest percentage, like 100% of cases, and degree of OCP pollution was found in group 5. Subgroup A, women living in the village Sakaldi near the former agricultural airport. It was mentioned in the picture where the local population were having the rest near the cropland. And the smallest percentage was in group 1, far from the former plague fossil, like 5.88 of cases. It was the distance about 6 to 7 kilometers from the former plague fossil and consumed food grown in their garden and obtained from domestic animals grazing away from the former plague fossil. So it means away from the areas polluted by OCPs. So therefore, the data obtained indirectly indicated that despite the fact that more than 30 years have passed, OCPs remained unchanged in the soil. So when we were conducting the seminars, webinars, meetings, conversation, and preparing the calendars, booklets, handouts, we used the result of scientific research, data on pathologies diagnosed by medical specialists, and laboratory tests. So we also prepared recommendations for the local population. Local authorities, agronomists, environmentalists, medical workers, member of different NGOs, means non-governmental organization, and activists. So nevertheless, based on the results of the study, the following recommendations can be made for the population living in the study area. So prohibit farmers from growing fruits, vegetables, legumes, gourds, fodder crops, as well as grazing animals at distance of minimum 4 to 5 kilometers from the polluted area. Means agro airports and destroyed pesticide storehouses. To state environmental protection agents and local agronomists are recommended to carry out soil remediation through winter spring soil leaching, planting trees, which are then used by local residents only as a building materials. For medical workers, the population living in the near place of former polluted areas to be classified as a risk group, and for women to conduct breast milk or breastfeeding examinations for OCP if they are found, to take appropriate preventive measures. So as regards scientific aspect, it's necessary to study the actions of OCP in the human body that penetrate through the GIT, taking into account the nature of diet and lifestyle of the local populations. Means ethnopsychology, what kind of fruits and vegetables they used to use for their diet, how they cook their dishes, which dishes are taken mostly in a raw or already fried or boiled. So all of these things should be took in attention. So also during the seminars, we cited specific examples. For example, your and your child's illness is due to OCP in your body, which came through cow's milk, meat and dairy, vegetables, melons, grains, and legumes grown in the polluted areas. So therefore, in order to maintain health, you should follow these recommendations. In these places and fields, you have to plant trees only for building materials and graze animals away from polluted areas. So such meetings and seminars gave good results. The local population began to comply with our recommendations and demand that the authorities solve environmental problems. So here you may see like outreach or the work with population should be started from the discussion and explanation to the most respected members of the society in the Central Asia, means aksakals. It's translated as an older generation. So in the society, they have a big authority, and if they come to the conclusion, the population will listen to them. So meeting of the physicians with the women. In order to improve, raise the awareness and achieve compliance with safety measures and implementation of recommendations by the population living in the places polluted by OCPs. So it's necessary to conduct a comprehensive study to determine the degree of danger for the population health. So for this, it's necessary to use breast milk as a biomarker and carry out a preventive examination of the population. So when carrying out activities, use the data obtained from studies with specific examples. Provide the local population the results of studies and recommendations so that they could demand from the local authorities. So the implementation of measures to eliminate OCP's pollution on the basis of the Stockholm Convention. To increase the effectiveness of ongoing seminars, meetings among the population, and to raise the awareness and executive discipline, it's recommended to conduct a comprehensive study of the area. Examine the local population by using our methods with the elements of evidence-based medicine. So that's all. Thank you for your attention. If you have any questions, please don't hesitate.
Thank you, Timur. Okay, then let me try to wrap up, I think, a very mixed set of various presentations. I think we heard from the last two speakers, obviously, that we have a legacy, and it's not visible only in landfills, but also in the soil on which we live and in the bodies which carry our life. And so it is a real issue, and we need to find real life solutions. So we have heard two presentations. I liked very much your quote saying that we should not only remediate the soil, but also we need to think how to remediate human bodies. So we have heard, I think, a very interesting option, both from you, again, supporting the Mediterranean diet, that it's very healthy. You should try to add the 1 glass of red wine, whether that also helps. And also from Rahmanbek, I thought it was a very interesting to hear what medical plants from Kyrgyzstan could do, and I hope there will be possibilities that this study could be pursued, and also, obviously also see whether that could be used on a wider scale. We have also seen a few presentations, obviously looking at the toxicology, but it's also clear that that a lot of things are still unclear, and it's not easy to say. And in my opinion, we should probably also start not to look at the traditional cause-impact relation, but today we are living in a world where there is not one single chemical impacting you and having one single cause. but we're having obviously a world where we are living in a mix of cocktails outside and inside of us. And as you probably know, I mean, there are discussions in the European Union, how could we measure that in a more meaningful way? A key word could be bioassays, which measure the impact of a cocktail of chemicals instead that you stay in the classical way of simply measuring the toxicity of a single component. And our first speaker obviously reminded us what also Mark already said yesterday in the Central Asian panel. We thought one day we are cleverer than nature, so we have used a lot of chemistry, and as I always say, people first do something and then they understand what they do. So we understand that we are not cleverer than nature, and we should probably try to go back to how nature has held everything into balance. But it's a long way. It's a lot of awareness raising, as we heard both from you, is needed in Spain, but also we heard in Kyrgyzstan, it's very important that people more understand all these interconnections. So there's a lot of work for us to do. And I think that's great, because I think we can do something meaningful in our lives. And so I would like to conclude this session with many thanks first of you for your interest, and the interest means you want to do something about it. So good luck in whatever is ahead in your lives. I don't know whether we will meet again, because at the moment, as far I know, this is the last forum, but maybe life brings us all together at some moment, and until then, all the best, good energy, creativity, and good advances in solutions. And many thanks, and good lunch now.